This is a production of Cornell University.
Good afternoon. It is hard to believe that this is the last time that I will be giving this talk. I've given a very similar talk over the last 3 years, and I got to call it the annual fossil seminar. And it looks like this will probably be the last one for a little while. So this is the final fossil seminar. So my work for the last few years has been on understanding angiosperm fossil taxa of the Eocene locality Laguna del Hunco in Patagonia, Argentina. The objectives of my research are to describe new Laguna del Hunco angiosperm taxa, put them in a phylogenetic context, and assess what this new information can tell us about our understanding of the locality and the biogeography of the relevant family or clade. But why does this research even matter? There are several biases in the fossil record, some that are difficult to mitigate, including the environment of deposition. How was this buried? Is this in a desert? It's a lot harder for it to bury than if it's in a lake. Was there oxygen to initiate decomposition? The organism being preserved. Bones and hard shells preserve a lot better than soft tissue. How close the time period is to current time. The further back in time you get, the more time has occurred that rocks can have been recycled, making it easier to find stuff that's closer to modern day. But one bias that's much easier to contend with is the geographical bias. This bias is rooted in several factors. First, how fossils are found. Vast untrekked wilderness, less likely to be able to find fossils. Political landscapes. We have all seen the front page news over the past 3 years. It's not safe for me to go do research in Ukraine. But the reality is, a lot of sites are currently at risk right now. And unfortunately, because of this political landscape, I just can't go look at them. Current biome. It's easier to find fossils in places that aren't covered in glaciers or massive rainforests, which weirdly makes paleontology one of the sciences that actually might benefit from climate change. And geological status. It's hard to get at fossils that are under the water or on top of a mountain. You might not know this, but Everest is actually covered in marine fossils, but trying to get up there is a little difficult. And access. Even if I wanted to, I can't go look for fossils in North Korea. I have found a total of 1 paper on North Korean fossils that had a non-North Korean author, and they were from China. So I don't think they're gonna let a US paleontologist go do work there. The result of this is that paleontological research for the 19th and 20th centuries were centered around the United States and Europe, with Asian paleontological research not becoming a global competitor until the 1990s, when China opened their paleontological research to the West. But what this means is that South America, Africa, Australia, Antarctica, and most of the oceanic islands are vastly understudied, leading to a lack of data points from these areas and the potential for biased or skewed results that might not accurately portray the range or biogeographic history of certain groups. Now, you might notice on the map that the group of understudied areas I listed encompasses the entire southern hemisphere, as well as most of what was the former Gondwanan landmass. So models are being made of paleoclimate estimates, sometimes not including any data points from the southern hemisphere, even though they do exist, although there are fewer of them. And this is a huge problem, as the southern hemisphere has its own set of ocean currents and air currents that aren't actually connected to the northern hemisphere. So this becomes a real problem because this is not being reflected in the modeling. To combat this, we need to do more high-quality research in the southern hemisphere collaborating with local institutions to promote the research being done there. And that's a big part of what my dissertation was about. My dissertation is split into 5 chapters. The first introducing you to Laguna del Hunco, then 3 new taxa from Laguna del Hunco, and the final chapter is on understanding the paleontological record with a narrative perspective. Laguna del Hunco, the site that we work on in collaboration with researchers from Penn State and the Museo Paleontologico Egidio Fruglio, a museum located in Trelew, most well known for their discovery of Patagotitan, which is the largest known dinosaur. Laguna del Hunco today is in the steppe of Patagonia. It's very, very dry. Pictured on the right is one of our rock splitting parties, as we like to call them. And if you were wondering how big the rock formations are, Alejo decided to pose, giving you a sense of scale. Laguna del Hunco is currently located in the Chubut province of Argentina. The caldera that makes up the site is about 10 kilometers by 30 kilometers, or about 300 square kilometers. In comparison, Crater Lake in Oregon is also a caldera, but it's only about 8 kilometers by 10 kilometers. 52 million years ago, Laguna del Hunco was about 5 degrees further south, which is about 350 miles. So if you went 350 miles straight south from where we are right now, you would be in southeastern Virginia. But it was also significantly further east. If you look at the image, you can see a very faint yellow outline just west of this image of South America 52 million years ago. That faint yellow outline is the modern day coastline, so it was quite a bit further east. Alongside this, the Drake Passage, which separates South America from Antarctica in modern day, had not opened. There was either a sliver of land or very shallow water connecting these 2 land masses. And the Isthmus of Panama was also wide open. So there was no land connection between North America and South America. Geologically, this was a very volcanically active area. The maximum age, based on uranium-lead isotope analysis of zircon crystals from the oldest layers of the site, is about 52.2 million years old. And there are various fossils deposited in the 170-meter tall fossiliferous zone that covers about 200,000 years of time. Now, as I said earlier, this is a caldera, specifically a trapdoor caldera. So repeated volcanic activity would collapse portions of the volcano in trapdoor-style collapse. So in this case, when you have the magma chamber erupt, you see that open space, and then it collapses, and you form this lake. Instead of collapsing straight down, Laguna del Hunco collapsed at an angle. That's why it's 30 kilometers by 10 kilometers instead of 30 kilometers by 30 kilometers. But this happened a couple of times. These eruptions created depressions that would often fill with water to make lakes. Again, we're going to look at Crater Lake just because it's a good example. This is Crater Lake in Oregon. This is actually much, much smaller than Laguna del Hunco's caldera. It's about a third of the size, but you can see entire mountains over here. It's still very large. And one of the things that's unique about this, that is something you see in caldera, is if you look at Wizard Island, that is the start of a new cinder cone volcano. So that is the start of a potential new eruption one day. And this is what was happening at Laguna del Hunco. You had these lakes form, and then you would have new cinder cones come up, erupt, and seal in the contents of the lake. Climate-wise, this was during the early Eocene climatic optimum, which is this region right here, with global temperatures around 15 degrees Celsius, or about 27 degrees Fahrenheit, higher than modern day. So you had alligators and palm trees hanging around up in Alaska at the same time. And for reference, 0 is where we are right now. That's the Paris Climate Agreement maximum. So it was quite a bit warmer than anything that we've seen in recent years. Paleofauna of the locality includes insects like scorpion flies, dragonflies, stink bugs, and weevils. There were also catfish, pipoid frogs, turtles, and birds. All of these support the interpretation of Laguna del Hunco being a rainforest freshwater lake. Fossil pollen and spores includes ferns from the families Dixoniaceae and Osmundaceae, along with gymnosperms like Araucaria and Podocarpus, and several angiosperm families including Juglandaceae, Malvaceae, and Nothofagaceae. With macrofossils, there are ferns like this Todia fern, which is part of the family Osmundaceae. Macrofossils of gymnosperms include Araucariaceae, Psychidaceae, and Podocarpaceae. Podocarps in particular tend to have very little drought tolerance, sticking mostly to very wet forests, which falls in line with other ecological interpretations of this locality. Macrofossils of angiosperms display a large range with fruits of Solanaceae and Juglandaceae, wood of Winteraceae, seeds of Menispermaceae, and various specimens of Monomaceae, Ripgoniaceae, which is a monocot, Aconiaceae, Myrtaceae, and the list keeps going. I could spend the next 5 minutes listing things. It is arguably the most diverse fossil Eocene flora in the Southern Hemisphere and among the most diverse in the world. Most of the species found so far, particularly among the angiosperms, have either a very globalized distribution or a very distinct biogeographical connection to Australasia, like the Myrtaceae, which proved to be the oldest eucalyptus in the world, completely upending previous hypotheses regarding the genus. Among the angiosperm fossils found at Laguna del Hunco are these 3 fossil taxa that I studied that belong to 3 separate families: Malvaceae, Malpighiaceae, and the order Phagales, with an unknown family. So starting with Malvaceae, Malvaceae is a very large family of flowering plants that includes 422 living genera and around 4,225 living species, distributed among 9 subfamilies in temperate and tropical regions around the globe. It is referred to as the mallow family and contains a lot of really important species, including cotton, okra, cacao for making chocolate, durian, hibiscus, and the origin of marshmallows, the original marshmallow, which, don't worry, is no longer an ingredient. So these fossils that we found at Laguna del Hunco are branching infructescences of Malvaceae fruits, particularly belonging to the subfamily Malvoideae, which has cotton, okra, hibiscus, and the original marshmallow. With the individual fruits, there is an extremely short pedicel, which is shown with the red arrow, and a subtending bract, which is shown with the blue. The fruit is a dehiscent schizocarp. It is a dehiscent schizocarp with 5 completely separated mericarps. These mericarps are ovate with an acute apex and a ridge down the middle of each. And it's okay if you don't know what a schizocarp is. I'm about to tell you. A schizocarp is a dry fruit that splits into single-seeded parts when ripe. It's kind of like the chocolate oranges that a lot of people get at Christmas, where you smack it or drop it and it breaks into those individual little wedges. But think of those individual wedges as a mericarp that has 1 seed. So in this case, our fruit would have 5 of these. We named the species Uherkaruan. The name translates to vessel of the old and comes from the language of the Telhuche, the indigenous population that inhabited large parts of eastern Patagonia. Although they likely did not inhabit Laguna del Hunco itself, their language is believed to be the closest to the Texian, the group which did inhabit the area, but unfortunately this group did not survive the Spanish invasion. The significance of this fossil is that this is the first fossil Malvoideae fruit found in either the Southern Hemisphere or the Americas. And it provides data as to the biogeographical path taken by the Malvoideae subfamily. The next fossil species came from the family Malpighiaceae. This family is much smaller than Malvaceae. It only has about 77 genera and about 1,300 species. These can be trees, shrubs, herbaceous, or vines, so it kind of runs the whole gamut. The phylogeny has fluctuated a lot over the past 25 years, with major changes happening as recently as May of 2024. The family is mostly known for their flowers, because their overall floral appearance stays rather consistent. Particularly among the neotropics, it has 5 sepals, 5 petals, glands under the sepals, 3 styles and stigmas, and all of the American species, both North and South America, produce oil. All of these similarities have led to fruit morphology playing a larger than average role in the identification of these species. Like Malvaceae that I showed before, the fossils that we have are a schizocarp of mericarps. But in this case, each one of those single-seeded chocolate wedges is instead one of these, a 4-winged mericarp. These mericarps have 4 spathulate wings that are generally around 4 to 6 millimeters in length, with 2 of the wings on each mericarp being 1/2 to 1 millimeter shorter than the other. Within Malpighiaceae, there are several accepted clades, one of which is the Hyptigi tribe. The tribe is composed of 17 genera and around 377 species. It has gone through several iterations of reconstruction in the past decade, including the creation of Glycophyllum in 2021 and another completely restructuring in 2024. Within Hyptigi, there are 3 genera that display the 4 lateral wing traits in some of their species: Tetrapterus, Glycophyllum, and Needhamzuella, all of which are exclusively neotropical, so only found in the tropics of the Americas. The 4-winged trait is hypothesized to be ancestral based on ancestral state reconstructions in the literature. There are subtle variations among these 3 genera, but species in these genera tend to be schizocarpic with spathulate wings and may have a dorsal winglet or ridge. So our fossils share a lot of commonalities with these 3 genera, and teasing them apart is a little bit difficult. But the biggest thing is that we see the traits that are all common of Hyptigi. We see these spathulate wings, and like Tetrapterus, we see that 2 are slightly larger than the other 2. The center of the fruit has some sort of debris around it. Now, they don't often in the literature show the back of individual mericarps or fruits, but behind these fruits, especially in herbarium specimens, you tend to see this amount of floral debris that gets left behind and kind of sticks around, which we think is what this haze is around that central globose structure. One thing of note is that in many herbarium samples, we see specifically the presence of a style remaining on the individual mericarps, which we do believe that we are seeing here. And we see the same thick pericarp wall, which is very imperative that only in Hyptigi, and I'll outline it in yellow for you, only in Hyptigi, but also in a couple other Malpighiaceae, you see this thick pericarpic wall producing the wings. That's something that's very specific to Malpighiaceae. And then we also see the same venation type where you have the veins are much denser towards the base, kind of spreading out as you get towards the margin. Bayesian analysis was conducted using Mr. Bayes. In repeated analyses, our fossil taxon and the fossil Tetrapterus harpiarium, shown in bold, appears as part of a polytomy with the other groups of Hyptigi, except for Glycophyllum, which is found sister to the rest. This might seem much less resolved than the maximum likelihood analysis from Almeida et al., but if you collapse down all the nodes in Almeida that have less than 50% support, which is our minimum threshold, then they start to look really similar and show the same polytomies. So we are getting essentially the same results as the DNA only, just with our added fossils. We assigned the species to the existing genus Tetractris and gave it the species name Dolgopole in honor of Matilda Dolgopole de Saez, who was the first woman to get a degree in paleontology in Argentina and the first female vertebrate paleontologist in all of Latin America. She also described and published new fossil species of fish from Laguna del Hunco in 1941. Now, the modern distribution of Malpighiaceae is tropical to subtropical, with most of the diversity being in the Americas. But when we start to look at the fossil record, this changes a bit. Looking at just the macrofossils, we have our fossil, which is the oldest down in South America, followed by the Eocene flowers from North America that was described by our own Bill Crepay. We have Oligocene fruits from Europe and much more recent Pliocene fruits from Asia. And this starts to paint a picture of this group starting out in South America, moving north, going across into Europe and Asia. And that's been proposed a lot. The problem is that that doesn't hold up when you include the pollen record. And a lot of people don't. But once you start to include the pollen record, you see pollen in South America popping up in the Eocene. In North America, you see it in mid to late Eocene or late Miocene. It's kind of staying the same so far. But then you get pollen in Africa in the late Eocene. And then by the time we have fossils in Europe, we have pollen in Australia. So it starts to paint this picture that what might actually be happening is a much more globalized distribution of this family by the Eocene that then contracted with late Cenozoic cooling to its modern distribution. So the significance of this fossil is that it's the first Malpighiaceae fossil fruit found in the Southern Hemisphere. It's the oldest Malpighiaceae found in the world. It expands the poor fossil record of Malpighiaceae. It's the first angiosperm fossil from Laguna del Hunco connected to an exclusively neotropical group. And when combined with existing data, suggests a more global distribution of Malpighiaceae that contracted from late Cenozoic cooling. The third fossil fruit belongs to the order Phagales. This order includes families like Fagaceae, the beeches and oaks, Juglundaceae, which includes walnuts and pecans, and Betulaceae, which includes the birches and hazelnuts. The fossils that we have are thyrsoid infructescences. The fruits are contained in a 5-valved cupule, and the cupule has an attached pedicel that has a slight flare at the base, suggesting natural senescence rather than breakage. Valve trichomes are long, sometimes twice the length of the valves, and these trichomes can be seen on both the margin and the abaxial side of the valves. If we take a closer look at the valve trichomes, we can see that they have a slight lean towards the apex. This is something that we see a lot in Phagales. The valves are irrigated by a midvein and 2 parallel marginal veins connected by transversal veins. The fruits within the cupule is a cluster of 5 nutlets. Label them there for you. And fortunately, we have one specimen that splits at the base of the nutlets with the cupule. So on the right, we have the bottom of the nutlets, and on the left, we have the base of the cupule. So it's split where they would have fit together. Sometimes these nutlets appear to have very fine hairs on them. However, their lack of consistent presence suggests that this might be a part of the ontogeny. And we are very lucky to have what appears to be an immature fruit in which the valves are enclosing the fruit and are covered by short, wavy, immature hairs. As I said, having multiple stages of the ontogeny is actually pretty rare in the fossil record and provides us with a unique opportunity to study this fruit's development. We also are lucky to have specimens with fruits still attached and fruits in the process of detaching, which allows us to see that the pedicel is part of the whole dispersal structure along with the cupule. So our theorized ontogeny is that the cupule develops enclosing the fruit and has short trichomes initially that then elongate and the valves open up. This is consistent with the process that we see in phagales a lot. The fruit inside the cupule is theorized to begin pubescent and lose those hairs as it matures. So it starts out fuzzy and then becomes kind of this slicker outer edge that you see very often in phagales, particularly Fagaceae. So we are putting this in a new genus and species. The genus name is proposed is Quechu, which is named in the Mapuche language. The Mapuche are a group of people native to southwestern Argentina and eastern Chile, known for their long-standing resistance to the Spanish colonization. Quechu is the Mapuche word for 5, referring to the part of the fruit and cupule being in sets of 5. The species we are naming Fratercis is a compound of frater, meaning cousin, and sus, meaning it, which is named for its resemblance to cousin it when you invert the fruit. The fact that the fossils of this species are found in such a large portion of the fossiliferous zone means that it was around for roughly 100,000 years at this one location. To have such a species with such an unusual morphological set of characters is a really unique opportunity to study not only the development of these fruits, but also what was there and for how long. Summarizing my work on the 3 fossil species, they support the hypothesis that Laguna del Hunco was likely a wet tropical forest, despite its latitude. My work continues to the tradition of Laguna del Hunco serving to push back origin dates and expand the paleo ranges of various family. It adds to the evidence of Laguna del Hunco being among the most diverse Eocene fossil sites in the world, and adds evidence of at least some of the species from Laguna del Hunco sharing a connection to the modern neotropics. But as Alejandra mentioned, and many of you know, I am also very passionate about science communication and science education. So as part of my dissertation, I have completed an outreach and education project called Fossil Fridays, which was formed out of the idea to use the narrative format to better convey the paleontological record by creating narrative reconstructions of specific fossil localities. Why the narrative form? The use of the narrative form to convey meaning has a long history, dating back thousands of years with its use as oral tradition. And you can find it in fables like The Tortoise and the Hare, which actually dates back to Aesop, an ancient Greek storyteller who may or may not have actually existed. They're a little dicey on whether or not he did. But in many of these stories, the lesson is really general, and the culturally relevant details that make the stories familiar to us can be swapped out to be adapted to whoever you're telling the story to, without changing the meaning. And this idea of the power of the narrative is backed up by science. It's backed up by these cognitive studies that indicate that the use of the narrative can increase information retention, persuasiveness, and elicitation of empathy. And it makes sense. Telling the story of one person's struggle feels more impactful than just giving you the stats. These narratives are sometimes strategic, used for good, such as bringing attention to a humanitarian cause, a crisis, or an emerging atrocity. Other times, these narratives are used as an extremist propaganda tool to reinforce an extremist ideology. Either way, the narrative has proven to be a useful tool. Within paleontology, though, the narrative is used, but almost all of the media that is reconstructions of ancient life is dominated by dinosaurs, with the occasional Pleistocene megafauna. It just keeps going. I didn't even include all of them. So if we look at the Cambrian forward, the Cambrian being when we start to really see complex life that we can show in these narratives, most of the examples of these narrative reconstructions have the T-Rex in them. Okay, the T-Rex was only alive for the last 2 million years of the Cretaceous. Add in, let's say, 13 million years to kind of cover some of the other dinosaurs that weren't around at the same time, and the 1.7 million years of the Pleistocene. Whew, that's not a lot. That's only about 3% of the Cambrian forward. So there is so much diversity that we're just completely skipping over in these narrative reconstructions. And that's where Fossil Fridays comes into play. The goal for the podcast was to produce environmental reconstructions of various points from the Cambrian forward in an easy to digest way, creating a brief overview for a fossil site that is understanding and engaging to reach the public, while also providing an introduction for scientists who are not familiar with paleontology to understand the environmental history of their study groups. And overall, just to show how amazing the fossil record is, because it's not necessarily shown to the public. To do this, each of the reconstructions follows a similar pattern. We start out with the listener arriving at the location in modern day. The purpose of this is to provide a direct comparison for when you step back in time, to give you a baseline of what that place in the world looks like right now. Then the listener steps back in time. Now, I've chosen not to use any sort of time machine or crazy thing to happen because those cognitive studies indicate that the more fictitious elements that you add in, the harder it is for people to understand what's fiction and what's fact and kind of parse those out. So by minimizing this to just they step back in time, I'm minimizing the fictitious elements to really focus on the facts and recreate these for people. Then the listener explores the environment. And this is using all of the senses. Sometimes you smell the rain or animal dung. Sometimes you feel how humid it is or how soft a petal is. You hear the noises that the animals are making. You see the landscape in front of you. And this serves to introduce them to not just the big megafauna that everybody knows about, but everything. Looking at the plants, looking at the animals, for everything from the tiniest of insects to the largest of trees, trying to really understand what it would be like to walk through that environment at that time. And then the listener encounters and overcomes some sort of conflict. Conflict is the crux of a good story. And sometimes these conflicts are really small. We all know exactly what it feels like to step into water or step into mud that you didn't think was going to be that deep. And the water goes into your shoe, and then your socks wet, and it feels gross and squishy. We all know what that feels like. So I wanted some that were relatable, and then there are some encounters that are less relatable. Things like being chased by Boverisuchus, a galloping crocodile. Yes, it could gallop, it's terrifying. That hopefully none of us have ever experienced, but it gives listeners that sense of adventure that you kind of expect from these sorts of things. So playing on this dichotomy allows people to put themselves in the situation, but also have that sense of adventure. And then the listener returns to modern day. And this serves to kind of wrap everything up and bring everything back to modern day, as well as giving me the chance to explain more about the site and things that I may not have been able to cover. This can include how do you see these fossils? How do you visit these places? More information for people who are interested. So how do we do? Well, for the first season, I created 12 full-length reconstructions along with various other tidbits. The reconstructions covered from the Pliocene, about 4.5 million years ago, stretching back to the Cambrian, 500 million years ago. Over season 1, I hit fossil sites in 6 out of the 7 continents. There were more in North and South America than other continents, but that was partially because I had personally visited 4 of the sites. So I was using my actual knowledge of the location and what it was like to walk through it in modern day. But that wasn't good enough. So for season 2, another 12 episodes, which these are labeled in black on this, I wanted to not just hit those 6 continents. I wanted to hit all 7. And we have. A third of the sites are in the Southern Hemisphere, with half of the sites being from understudied areas. So in summary, this podcast, which has been probably 2 years of my life now, has covered 17 fossil locations. I've done 19 episodes of reconstructions, 2 special episodes, 10 short topic episodes, and created over 10 hours of content, which is longer than the theatrical cut of Lord of the Rings, but shorter than the extended edition. I was going to use Marvel, but it was like way longer than the Marvel Infinity Wars, so we went with Lord of the Rings. And of course, I have so very many acknowledgements to make at the end of this. It's been a long 4.5 years, and I have a lot of acknowledgement slides. Starting with institutions and funding, I want to especially thank the MEF. They were wonderful hosts down in Argentina, and I made so many friends that hopefully I will continue to collaborate with. The American Philosophical Society and the Einaudi Center for paying for me to go down to Argentina, which is very important, as well as the Paleontological Society and AGU, who paid for me to go lobby Congress and learn about that process. Technical help included a lot of herbarium staff, not only from our herbarium, but also from the Harvard Herbaria, the US National Herbarium, and the Field Museum Herbarium. Phylogenetic support is mostly Jacob Landis. I showed up at his office a lot. His door is directly across from mine, so he couldn't escape. And don't worry, I baked him cookies a couple times as a thank you. There are several people who have provided mentorship, whether they know it or not, and that includes Bill Crepay, Hannah Marks, and Margaret Frank. I've asked them loads of questions, and they've been more than happy to help, even if they weren't official mentors. Fossil Friday support, Patty Leva designed the logo. This would not have been possible without that. I can't imagine that my drawing skills are anywhere as good. And Greg has always been, my lovely husband, Greg Couch, has been the executive producer, always making sure that the website stays up to date and the bills are paid on that. I also want to thank lab members Jennifer Svitko and Noah Crook, who have been instrumental to this. And just like a village, a PhD dissertation takes a community. I want to thank the Naughty Knitters trivia team, the lunch group that I've eaten with for the past 4.5 years, the Big Red Barn knitting group, the plant biology graduate students, and of course my family. And then the 2 creatures that this would not have been possible without, my guinea pigs. I can feel Bill rolling his eyes right now. And we got raised shortly after we moved here, and they've really just been there for all of it, sometimes doing things they shouldn't, like climbing onto my laptop and trying to type. And then, of course, my committee. If you don't know where this graphic comes from, last year I gave my grad student department presentation on Halloween, and I dressed up as Ms. Frizzles. Only one person from my committee showed up. So we're reusing the graphic. So starting from the cool kids at the back of the bus, Liz Hermson, as Alejandra said, like Liz is really the one who got me into this. This wouldn't have been possible without her. I never would have found paleobotany. She actually has known me longer by about 4 days than anybody in this room that is not a blood relation to me. Chelsea Specht, a big thank you to her. She really jumped in after my A exam when I had a vacancy on my committee. She just jumped in, was very cool about everything, and just was ready to go. Kevin Nixon, who has always been there to make me laugh and give really solid advice, honestly, on phylogenetics and everything else. And of course, Alejandra Gandolfo, who I truly could not have asked for a better advisor. And I truly mean that. She has been incredible this whole time, giving me just the best advice, backing off when I needed space, making sure she was there when I needed her, and really just tailoring her mentorship to whatever I needed. And I could not have asked for anything beyond that. And then the final person who moved 4 hours away from his friends and family and his job so that I could pursue my dream is my incredible husband, Greg, who is here. And I really could not have done this without him, from long nights and working weekends, showing up with pizza when I was working late in the lab and I was on a roll. I would text him and he'd be like, all right, it's 8 o'clock. I'll just roll up with pizza and work alongside you. And that got a lot of this done. So thank you so much to all of these people and all of you.
On the line.
Any questions?
And yes, any questions for Caroline?
Okay, so the cousin it plant,
Yes.
you were very fortunate to catch it at different stages
Yes.
of development. Okay, very general question. What about that site do you think made it that you were able to catch that?
So I think part of it is, one, because it's a thyrsoid infructescence, we're wondering if it developmentally was going from bottom to top with how it matures. So that may have allowed us to catch those multiple stages. But then also because this species was around for such a large chunk of the repeated collection and the repeated the site preserved everything, that we were able to get enough chunks to be able to catch those moments. So it's not just one event that sealed everything in, it's repeated ones over 200,000 years. And for 100,000 years, that species was there.
170 meters.
Yeah, it's big.
It's a little bit taller than even us.
Yes.
Is that typical of Juglandaceae or anything, or is there, I mean, are there other groups that are like that with that kind of longevity as in fossil records, or is this really unusual to have that?
Oh, no. So like the average lifespan of a species is pretty long. I mean, humans have been around for 300,000 years. Like the average lifespan of an individual species, I think, is around like 2 million. So that's not that unusual. What's unusual is that we were able to catch it at the same location for so many snapshots.
Do we have other known fruit from that location, and are those any of those fleshy? Because it give you indication that there were fleshy fruit there. I know it was.
So we do have a lot of fruits from that location. I'm trying to think if we have any that are specifically fleshy.
But the flesh probably wouldn't preserve, but you.
No, but sometimes with things like endocarps, you can get those. Like that is something that happens a lot in the fossil record. I'm just trying to think if there are any specific fleshy ones.
Oh, fleshy, we have the tomatillo.
Oh yeah, so we have like the solanaceae, but it's we have like that papery portion that goes outside of it
But you.
on the tomatillo.
The reason I mention that is because if I hear about a dried dehiscent fruit, it's not 100%, but I think arid climate. I think because usually that moisture drives evolution toward making fleshy fruit instead of dry. So it'd be kind of counter to the narrative here as a building. But if there is indication that more broadly there was fleshy fruit there.
We do have other stuff.
Yeah.
We have the oldest record of Euphorbiaceae, oldest record of Casuarinaceae, oldest record of I describe it, Mutaceae. So yes, there's tons of things.
Yeah, so some are fleshy, some are not. Fleshy fruits are slightly less likely to fossilize. With those, you're more likely to get the fruits and seeds. So I showed Menispermaceae. Oh, it's not going to show it. There we go. So this Menispermaceae, that's a seed of a fleshy fruit. So you won't necessarily get the flesh, but you will get the seeds, the endocarps, other things like that. So in finding those seeds, if they have something really distinctive, yes, we are able to find evidence of those fleshy fruits.
Do you happen to have any other tissues for those 3 species you focused on that have been preserved in that area?
Not that we know of. So the issue with this is that unless we find them directly attached, we can't prove that it's the same species.
Okay.
So we have maybe some leaves that might belong to the same family, but unless we find them attached, we can't prove that it's the same species.
With your podcast, what kind of audience do you get for that? And do you have any sense of like numbers and how broad it is?
So mostly from the US. I have had a couple listeners from Europe, mostly after IOPC, which was in Prague. I got some listeners from Germany after that. It targets middle school and up. I do know that my distant cousin shows it to his 7-year-old. So there's at least one 7-year-old listening to it. And then Jen is probably my most avid listener. And she got,
My bus driver who's
Yeah.
interested in paleo. It's like he was just driving and he was like, well, what do you do in your job? And I was like, well, I work at a paleobotany. And I just gave him a whole bunch of stuff to just look into. And I said, this thing, Fossil Fridays, go there. She does. And the next time I came back, the next time I got on the bus with him, he was like, oh, you know, I gave it to another friend of mine and we both listened to it and we had some suggestions. So, yeah.
So bus drivers also listen, apparently. Yes.
Is that correct that all of the fossils were zygomorphic flowers? Is that true?
Well, they were all fruits. We didn't have any flowers. No, we didn't have any flowers. It was all fruits.
But we do have flowers that are not psychomorphic.
Yes.
You also have a 5-year-old, my son, but anyway, who's he grabbing on to? Do we have any indication of who may have been dispersing this? Have you cross-referenced this with vertebrate fossils to see who may have been dispersing these fruit?
So most of the vertebrate fossils that we have are from the lake itself. So the other thing we found was a bird, but it's just like a hind limb, so we don't really know. Based on the structure of the fruits, we can make some guesses for some of them. So like the winged merocarps, probably wind dispersal. And then for the phagales, we actually aren't 100% sure, but based on the structure and those really long trichomes, we suspect that it couldn't have been like necessarily wind dispersal. So we have a couple of theories on that one. Maybe the trichomes hooked onto something, we don't know. But it would be really interesting. And I would love if we maybe one day found like some proto dog running around with one attached. That would be a great paper. Yes.
Do you have any sense for like what ecological niche these were? Like, were these actually aquatic plants, or were they on the shorelines next to the lake, or were they a little bit more inland in a drier setting? Do you have any idea?
So based on the family that they belong to and kind of the niche that those families tend to be part of, we suspect that these were near shore plants that either kind of just fell into the lake, or sometimes it's known for this location that there were slumps where plants that had grown on the ash previously, the whole like side of the lake would just fall in. So we do know that that happened a couple times based on some other research. So it could have happened that way as well. But yeah, mostly near shore or very close by, but not aquatic.
Well, thank you so much.
This has been a production of Cornell University, on the web at cornell.edu.